ready to take this cell through meiosis instead of mitosis. So now we still have our diploid cell, 4 chromosomes, 2 homologous pairs, but now this is a germ cell, and these are the only cells in the body that would go through meiosis to produce those haploid gametes. So this is a diploid germ cell. Now I'm not going to show all the parts of interphase. Again, I'm just going to summarize it and state interphase. But just like in mitosis that we talked about, it'll go through G1, where the cell will physically get bigger, and then it'll go through S to duplicate all of our chromosomes. So our chromosome 1 from the sperm gets copied, 1 from the egg gets a copy, 2 from the sperm copies itself, and 2 from our egg makes a copy. So we have those duplicated chromosomes, and we went through G2, so our centrosomes have replicated and divided. They're separating to opposite sides, forming those microtubules. All right, so now the good news is that prophase, metaphase, anaphase, and telophase are pretty well the same as what we talked about in mitosis, but we need to add a couple extra little things with meiosis. Also, we go through those twice, so we have a prophase 1 and then a prophase 2. So we also need to indicate them with a number. All right, so first of all, we're going to go through prophase 1. And similar to mitosis, the nucleus is going to break down. The microtubules are going to form that mitotic spindle. Our chromosomes are going to condense and get thicker, but the addition here with meiosis is that in prophase 1, we also get synapsis. And that's where the homologous chromosomes pair together. So chromosome 1 from the egg and 1 from the sperm will literally join together. Same thing with chromosome 2. Right? Again, they're condensing, so I'll draw them a little shorter and thicker. But again, chromosome 1 from the egg and 1 from the sperm are physically going to bind together in this synapsis. And the same thing happens with our chromosome 2. Okay, so this is our synapsis, where those homologous chromosomes physically pair together. So a microtubule from the left is going to attach to one of the homologous chromosomes. Microtubule from the right would attach to the other one. Okay, then a microtubule is going to attach to one of our homologous chromosome number 2s. The one from the left would attach to the opposite. Okay, so our sister chromatids, they're not important just yet. Here we want to separate our homologous chromosomes. So first they have to pair together and join in this event called synapsis. This is also where we could get crossing over occurring. Okay, and we'll come back. We'll do one more video that looks at that crossing over. All right, so we have after our prophase 1, now we're ready for metaphase 1. And here, again, we're going to get that tug of war, and we're going to line them up in the middle. So again, just like metaphase and mitosis, we're lining everybody up right in the middle. That's not quite in the middle, but imagine it being perfectly aligned right in the middle of our cell. Okay, so now we are going to go through anaphase 1. And now instead of separating sister chromatids, we are going to separate homologous chromosomes. So that synapsis event is going to break apart so that on each side of the cell, we only have one of each chromosome. Okay, so we have separated homologous chromosomes. And that's the main purpose of meiosis 1, is to separate mom or dad's chromosomes. So for each chromosome, we have one or the other, but not both. Then in telophase 1, we're going to form the nucleus around both of these clusters, and those chromosomes are going to kind of unwind and decondense. Okay, so at this point, we now have haploid nuclei. So we have two nuclei, but they're haploid. They have chromosome 1, in this case from the sperm, chromosome 1 here from the egg. But they're not homologous anymore. We only have one chromosome 1 in each nucleus. And then we had, for chromosome 2, it was reversed in this scenario. So where chromosome 2 from the egg came over here, and chromosome 2 from the sperm is in this side. So again, these are haploid nuclei. But they are still duplicated. Okay, so now we need to go through cytokinesis and divide this cell into two. Okay, so this one has chromosome 1 from the sperm and chromosome 2 from the egg, and this one's the opposite. It has chromosome 1, the shorter one, from the egg, and chromosome 2 from the sperm. So again, we've kind of mixed up our combinations, so we're not identical to either parent, but even if that did happen, where we had chromosome 1 and 2 both from the sperm, due to this crossing over, we would still have some genetic variability there. So again, the way that these guys line up in metaphase is completely random. So that independent assortment, these, both of the chromosomes from the sperm could be on one side, both of the egg on one side, or we can get this mixture like I've drawn here. That is completely random, and that leads to more variability through that independent assortment. So they can line up however they want to there. And again, we have 23 pairs of chromosomes, so the possibility of all of the ones from the sperm going on one side and all of the ones from the egg going to the other side, pretty unlikely. But again, even with that, crossing over would allow some more variability there. All right, so back to our stages. So we went through cytokinesis. Again, we have haploid cells, one of each chromosome, but they're still duplicated. So the second round of division is going to separate our sister chromatids, just like we did in mitosis. Right? So both of these cells are now going to go through prophase 2. We don't go through interphase again. We already duplicated our chromosomes. We don't need to do it again. So we go straight into the second round of division. Okay, so prophase now is just like mitosis. In both of these cells, the nucleus breaks down, the mitotic spindle forms, and our chromosomes are going to condense. Okay, and they're going to attach to microtubules from opposite sides, and the same thing happens in our other cell. Okay, so we've attached to our sister chromatids, they've condensed, and we've gotten rid of the nucleus. So now we're ready for metaphase. And now they're going to line up in the middle, just like they had done before. So again, that tug of war is going to occur until we get our chromosomes lined up right in the middle. And now we're ready for them to separate. So we line them up in the middle, and then both of them are going to go through anaphase 2. And now we're going to separate those sister chromatids. So again, this is still occurring in both cells, but that centromere lets go, and our sister chromatids are going to separate. So now we have separated sister chromatids. So meiosis 1 separates homologous chromosomes. Meiosis 2, the next round of division, separates our sister chromatids, much like mitosis. Right, now for telophase 2, now we're going to form the nuclei around all of our clusters of chromosomes. Those chromosomes are going to relax a little bit. They won't be so condensed. So we have chromosome 1 from the sperm here, chromosome 2 from the egg, and then in our other cell we have the opposite. Chromosome 2 from the sperm and chromosome 1 from the egg. Okay, so now they are haploid and no longer duplicated. So now we can go through cytokinesis again to give us 4 cells. Two of them are going to have chromosome 1 that came from the sperm. 2 of them will have chromosome 1 that came from the egg. So again, haploid, only 1 chromosome 1. And then chromosome 2 in these guys would have been from the egg. Chromosome 2 over here would have been from the sperm. So now we have 4 non-identical haploid gametes. So if this is a female, these would be egg cells. If this was a male, they would be 4 sperm cells. But due to crossing over and that independent assortment, again, with our 23 pairs of chromosomes, we can have a huge variety of different egg and sperm cells produced. And then, of course, which one actually comes together during fertilization to produce our zygote is completely random as well. All right, have a good day.